Hi, I'm Barnaby Clark, clinical scientist based at King's College Hospital. I'm the laboratory lead for precision medicine and responsible for delivery of genomic testing. We've got gene panels in service delivering myeloid disorders, lymphoid disorders, adult neurological brain tumours and paediatric tumours gene panels as well. I first heard about Kaisig technology at a QIAGEN user meeting. I'd previously been using the clinically relevant cancer panel. The Kaisig technology gave us, if you like, the best of both worlds. It gave us the depth and focused and targeted ability of the PCR amplicon panels, but it was also a library prep methodology which was far more simplistic than the bait capture technologies. So we thought that it would probably be much easier to kind of automate, because we previously had issues with automation. And prior to actually using Kaisig technology, we did look at evenness of coverage across our regions of interest, particularly in GC-rich regions, such as in CEBP alpha gene. We compared this to bait capture technologies and found that there's effectively about 1 log order better evenness of coverage that comes from the Kaisig technology. And this is really useful because you're trying to use that evenness of coverage. It means that you're really maximizing the sequence that comes off of the Illumina sequences to your regions of interest. So we've designed a very small 100 amplicon human identification panel in collaboration with QIAGEN. We're using Kaisig panels for MRD monitoring by sequencing. We then, using the diagnostic gene panel data, we can identify mutations that we want to use as markers to monitor these patients going longer term. We are restricting the region of interest down specifically to the actual patient and the patient's tumour profile that they had, and then we're sequencing a smaller region of interest per patient so we can maximise the amount of sequencing depth and reduce the cost. So we're hoping to be able to sequence on a MiSeq sequencer 8 to 10 individual patients with redepths up to something like 20,000, hoping to get to below 1% allele frequency detection. This is still a research project, but at the moment the data is looking promising and we're quite hopeful. We're looking at Kaisig targeted probe chemistry and its application for MRD monitoring using sequencing, and also to see if we can develop a non-invasive prenatal test for sickle cell disease. Firstly, the hands-on time of the library prep is down to around 6 hours, which means that we can do a library prep for a gene panel within a day, and then the next day we can quite comfortably kind of normalize and load onto the sequencer, and potentially with an extended day we could potentially deliver this in a whole day. So that's very attractive for cancer labs where turnaround times are short. The other part of the chemistry that we like is the minimal number of bead cleanups that are required in the library prep. Instead of having bead cleanups, it's enzymatically treated at every stage in the library prep, which means that the number of starting molecules within there stays the same. So your complexity of your library should, in theory, be better. And this is important in applications where you've got very few template starting molecules, such as in MRD monitoring or NIPT. But really the coverage is excellent across Kaisig. And also the other advantage of the technology being that it moves us into Build 38. And there's a transition at the moment going on in our lab of moving all our gene panels from Build 37 to Build 38. So I think for our sensitive assays for monitoring, it may be the sequencing chemistry of choice for us at the moment. I guess working with QIAGEN benefits us because it enables us really to kind of deliver our projects in as quick a time as possible. I've been working with QIAGEN really for my entire professional life, starting off with DNA library preps and DNA extraction, and that's continued through really to today.
QIAGEN.
Sample to insight.